So first of all, good afternoon, everyone. My name is Jingjing Kip. I'm an associate professor from the Department of Biological Sciences at DePaul University. I serve as the chair of the William J. Degutis Women in Science and Health Lecture Series Committee. On behalf of the committee, I'd like to extend a hearty welcome to you all, and thank you all for joining us for the fourth annual event of this lecture series. So as you may know, this is a hybrid event, and we have probably over 100 people registered for this event, and some of you are here in person, and some of you are attending virtually. So before we get started, I'd like to just remind the online participants that you may submit questions at any time through the chat function. And for in-person participants, you will get a chance to ask questions during our Q&A session. You may ask directly, just raise your hand. I will run the Q&A session too. So another thing is that this presentation is recorded, and the recording will be posted on the YouTube channel of our college once it's ready. Now I'd like to start today's lecture with a brief opening remarks. So we will hear more about the purpose and the importance of this lecture series from Dean Dansbarnes shortly. So I'm not going to spend much time on that myself, but we launched this the first lecture of this series in 2021. That was during COVID time, and that lecture was 100% online. We had great attendance through the virtual event. Since then, this lecture series has been held in every spring around this time with great success. And this year's lecture is unique, as this time the keynote speaker, Margaret Amsler, also known as Maggie Amsler, is an alumna of our own. And give a more detailed introduction to Maggie just a little bit later, but here I'd like to take this opportunity to thank Maggie for being here today to speak to us. Maggie, it's a great honor for us to have you here. Thank you. The William J. Degutis Women in Science and Health Lecture Series was made possible by the generous endowment from Dr. Linda Degutis. And she's an alumna of our CSH College as well. And we have also received strong support from the College of Science and Health, and also from the Provost office in all these years. And Dr. Degutis won't be able to join us today, but she has sent us a written statement, and later on we will read her statement to everyone. And we do have a dean of CSH joining here today, Dr. Stephanie Dansbarnes. And we want to express our great gratitude towards Drs. Degutis and Dr. Dansbarnes for your support. I know that Provost won't be able to make it here today due to some last minute arrangement, but she has been a strong supporter of this event throughout these 4 years. So thank you, Provost Gannon as well. As you may imagine, planning an event like this requires a lot of work. So here I also want to take this opportunity to thank the organizing committee for putting this event together. So you can see the committee members from this slide. We have Bernhard McWhinnie, Ty Ferguson, Blair Jennings, Dina Hader, Dorothy Kozlowski, Susan McMahon, and Damien Rudzinski. Did I say that right? Damien, sorry. So these are the committee members, and they have been helping a lot with this event. So next, we're going to have Dr. Stephanie Dansbarnes, Dean of the College of Science and Health, to give us introduction to this lecture series, and also talk a little bit about Dr. Linda Degutis. And Dr. Dansbarnes is an expert in cell biology and STEM education. She has been serving as the Dean of CSH since 2020. So now, Dean Dansbarnes.
Good afternoon, everyone. Thank you for being here today. And Jingjing has already mentioned that Provost Ghanem is so disappointed she couldn't be here today. She called me and she was really torn because it's just so much happening right now and she was needed in other spaces and places. And so I told her we would give her grace today. And so she wants to send her well wishes for this event because she has been a tremendous supporter. But I do want to welcome you all to the annual William J. Degutis Women in Science and Health Lecture Series. It is my honor to stand before you today, celebrating the remarkable achievements of women in the natural and health sciences and related fields. The series stands as a testament to the enduring legacy of Dr. Linda Degutis, whose unwavering support has made this event possible. Linda is a proud alumna of DePaul University, as Jingjing mentioned, and has not only excelled in her own career, but has also been a national beacon for policy, advocacy, and initiatives focusing on the prevention of violence, injury, and suicide. Her dedication to public service, rooted in the values instilled by her father, William, serves as an inspiration to us all. We are deeply grateful for her generosity and commitment to fostering scholarship and community within the college and beyond. Today, we are privileged to have Margaret Amsler as our distinguished speaker. Her journey in Antarctic studies embodies the spirit of exploration and discovery that drives scientific inquiry. As a trailblazing DePaul alumna, as we've all heard, she has not only expanded the boundaries of scientific knowledge, but has also inspired countless individuals, especially women, to pursue their passion fearlessly. From her undergraduate studies here at DePaul to her groundbreaking research in Antarctica, she exemplifies the spirit of innovation and resilience that defines our university. Her presence here today also underscores the invaluable contributions of women in science. As we listen to her insights and experiences, let us reflect on the countless women who have blazed trails, shattered barriers, and enriched our understanding of the world. Their resilience, intellect, and passion serve as beacons of hope and inspiration for future generation of scientists and health professionals. I would like to extend my heartfelt thanks to the organizers and planners who have worked tirelessly to make this event possible. Your dedication and vision have created a platform for dialogue, collaboration, and discovery. Together, we honor the legacy of William J. Degutis and celebrate the remarkable achievements of women in science and health. In closing, I do invite you to join me in welcoming our speaker as she comes up in a bit, and the spirit of inquiry and innovation that defines our community. May today's lecture inspire us to reach greater heights and forge a future where all voices are heard and all contributions are valued. And I want to take this opportunity to invite Blair Janice up here because she's going to provide some remarks on behalf of Linda, who couldn't be with us today. Thank you so much, Blair.
Yeah, thank you, Dean Barnes, and thank you all so much for being here. I want to share a bit of greetings from Linda Degutis, who is a wonderful alum and a trailblazer in her own right in the fields of science and health. She would like to extend thanks to the committee involved in this lecture series to invite renowned women scientists to present this lecture, and to Dean Barnes for her leadership. She wishes that she could be here with us in person to hear from a fellow DePaul alum, Maggie Amsler, who has contributed a great deal to our understanding of the Antarctic, and whose mentor and DePaul faculty member, the late Dr. Mary Alice McWhinnie, was the first woman to be named as chief scientist at McMurdo Station. Seeing women continue to explore new frontiers in science, to take their passions for learning and exploring to new horizons are exactly what the foundational concepts of this William J. Degutis lecture series and the values that her father instilled in her are so meaningful. Maggie continues to break new ground and to exemplify the importance of science. She is clearly passionate about and committed to her work and demonstrates the impact of a DePaul education. And after spending the day with her, I could not agree more wholeheartedly with that Maggie is an absolute exemplification of what it means to be a DePaul alum in service of our community. So we are so lucky. Linda says she can only imagine what she will continue to accomplish in the future, and hope that you will be inspired by her work in the lecture today. Thank you all so much for attending the lecture and for the work that you are doing to continue to ensure the importance of science in serving our communities. So without further ado, I'll introduce Maggie Amsler. Please give her a warm welcome.
Okay, well, thank you, Dr. Barnes, for the introduction, and thank you, Blair, for bringing us Dr. Degutis' wonderful statement. It's always inspiring to hear from both, and knowing that we got strong support from our college for women in science, and got strong support from our alumna to our scientific community here. So before we bring our keynote speaker here, I'd like to give a little bit more introduction to her. I remember last fall when the committee met, tried to decide which speaker to bring in for 2024 annual event. Maggie Amsler's name was brought to us, and then we were forwarded with the link to her website and with some other additional information. We looked through this information, and almost immediately everybody said, this is it. We got to bring Maggie here for our this keynote, to be our keynote speaker. So Amsler herself indeed is part of the Antarctica's history. As you probably know by now, Amsler earned her Bachelor of Science degree from the Department of Biological Sciences at DePaul University. Her mentor at DePaul at that time was the late Dr. Mary Alice McWhinnie. Amsler's first trip was actually as part of McWhinnie's team. She then pursued her master's degree and continued her Arctic studies at University of North Carolina at Wilmington. And then a career move happened, brought her and her husband, Chuck Amsler, a fellow researcher, to the University of California, Santa Barbara. At University of California, Amsler worked as a staff research associate and participated in an additional 10 expeditions in Antarctica. In 1985, Amsler was on the first ever US-sponsored winter cruise along the Antarctic Peninsula. She was aboard an icebreaker, the research vessel named the Polar Duke, collaborating with researchers at the University of California at Santa Barbara. In 2017, Amsler was on one of the first ever cruises using manned submersibles to document seafloor communities in Antarctica. She likely became the first woman ever to make a submersible submersible dive there. So in 2021, Amsler's adventurous scientific spirit was recognized by the Explorer Club, naming her the inaugural EC50 cohort, which represent 50 people the world should know about. Amsler's work during her first 12 expeditions to Antarctica followed the same path as her mentor, Dr. McWhinnie's, and she studied krill, which I'm sure she will mention that to you later. We had a conversation prior to this lecture. It's a very interesting creature. It's a shrimp-like creature, and the basis of Antarctic oceanic food web. Since trip 13, number 13, she has often worked alongside her husband, UAB biologist Chuck Amsler, and her boss, UAB biologist Jim McClintock. So initially studying the chemical defenses of marine plants and animals, but more recently also the impact of ocean acidification on calcifying organisms and the distribution of deep sea crabs that may be invading shallow waters as a consequence of ocean warming. Because of their numerous contributions to Antarctic science since 1980, in 2007, Maggie and Chuck Amsler were honored by the U.S. Board of Geographic Names with the designation of Amsler Island, which is about half mile away from the Palmer Station. That was the station that she worked together. She went on for her first trip with Dr. McWhinnie from DePaul, which started her career. So I just, I don't know if maybe Maggie will show you a picture later, but I did a little bit Google search and located this island named after Maggie and her husband, Chuck. So keep this in mind, it could be a next vacation destination, right? So Amsler has been author or co-author with her polar colleagues over 50 publications. In 2022, Amsler was named a Sigma Xi Fellow. So as some of you may know, Sigma Xi Society is a very prestigious scientific society. So it's such a great honor for anyone to be included as a Sigma Xi Fellow. There are also some fun fact about about Amsler. So most of her Antarctic fieldwork has involved scuba diving, and she has logged, guess how many times, dived in the icy cold water. She has logged 500 dives in the icy water, not just for fun, but to study krill, chemical ecology, and the ocean acidification. And when she is not in the water or on the waters, she will be, you can find her doing yoga or bicycling in the small gym. And when possible, she will do the cross-country skiing on the glacier. And at night, she enjoys sleeping out in her bivy bag under the Antarctic sky. Imagine that. What a life. Amazing. So it's my great pleasure to introduce Maggie to this lecture. And let's see the title here. Here, this is and the title for her today's talk is on this slide, Antarctica's Marine Realms from the Shallow Subtidal to the Deep Sea. Maggie, please come.
Well, I'm actually, at the moment. Thank you for all the marvelous introductions, and thank you especially to my hosts for the day. It's really been a joy to be back on campus, even though about the only thing I recognized was the pit in SAC. It's really wonderful to see all the changes and the growth and all the great stuff happening here at DePaul University. Makes me want to come back here and be a student. Well, my plan for today, if I can remember half of what I'm supposed to talk about at this point, is to, as you've heard and read my title, I'll talk about the shallow subtidal to the deep sea. And by the shallow subtidal, I mean that area just below the lowest low tide. So this diagram is in the same perspective as that opening slide, and it indicates the other major continents for orientation. The finger-like extension that points up from the continent is the Antarctic Peninsula, and it's pointing towards South America. And Andros Island is highlighted here because it's Andros Island that is the home of Palmer Station, and Palmer Station has been my home away from home during my time on the ice, as we say, when we're in Antarctica. The green dotted line represents the Antarctic Circle, and that's at 66 degrees south, and points south of the circle have 24 hours daylight on the shortest day, 24 hours daylight on the shortest day of the winter. So in Antarctica, that's June, the June solstice. Conversely, 24 hours of sunlight on the summer solstice, and in Antarctica, that's December. This really should be a lot easier for me because I tend to think backwards to begin with. So we're in the southern hemisphere, everything's kind of backwards. So Antarctica is a very light-driven, cold continent, and it's even more light-driven and generally colder in the water. And we're going to spend the majority of our time in the water. But I want to give you a couple introductory slides, including this next one. During the winter time, when the air temps are colder and the weather is generally calmer and less windy, the majority of the Southern Ocean the surface waters can freeze and form what's called annual sea ice. So all the white around the great continent here indicates ice, and that ice can almost double the area of the continent in the wintertime. And any of that, of course, and a layer of ice on top of seawater is even going to impede any available sunlight even more for organisms in the water column that need light. But basically, Antarctica is just a continent of nothing but snow and ice on the surface, and it is surrounded by frigid water. And if you really want to get to know Antarctica, you need to go underwater. So let's just dive right in. Well, after a stop at Palmer Station. So this is what Palmer looked like when Team DePaul arrived in January 1980. Palmer is the smallest of 3 U.S. bases maintained by the National Science Foundation. At peak maximum, there's 44 people. So you get to know people pretty well on station. The ship in the foreground was our transport from the southern tip of South America across the Drake Passage. It's the much storied 125-foot wooden-hulled vessel called the Hero. The station directly outside the back door of the station, if you will, rises a glacier that pretty much covers all of Andros Island. And the glacier is in this, oops, no, I was going to press the wrong button. In this image is quite close to station. Nowadays it's it's receded quite a bit. The ice floating in the water around the Hero is some of that annual sea ice I just talked about that hasn't quite broken up throughout from the winter development in the wintertime. And what brought Team DePaul to Antarctica is this critter, the Antarctic krill Euphausia superba. It's a scientific name. And it's commonly referred to as or described as a shrimp-like crustacean. So crustaceans are a class of arthropods, and arthropods are spiders, crabs, lobsters, shrimp are probably the more common. Arthropods that are also crustaceans. All crustaceans have in common, they're all generally aquatic, all have a hard exoskeleton, lots of segments, and jointed appendages. The word krill is a Norwegian term, which means little fish. And it's probably applied to krill because krill are sort of like fish in that they're pelagic or swimming all the time, just like fish. And they also tend to form in schools, like fish will school. You can find krill in all the world's oceans. You can only find this species, Euphausia superba, in the Southern Ocean, hence its common name, Antarctic krill. This particular species, I think the epithet superba is very appropriate and describes the fact that it's the largest of any of the congeners. It can be about 2 inches long as a fully grown adult. They're also found in huge schools, massive swarms. I also think they're called, that the specific epithet superba is very appropriate because they're superbly adapted to this really harsh environment that is limited by light or governed by light and cold. And also I think they're just superbly beautiful. What's not to love about a creature that looks like that? But krill are also really challenged in that harsh environment. They're really high in protein, and that makes them, coupled with the fact that they're very abundant and an easy prey item because of that abundance, they're under really high predation pressure. So it's hard to give an Antarctic marine talk and not talk about the national bird of Antarctica, the penguin. This is the most common species of penguin along the Antarctic Peninsula. It's called an Adelie penguin. Adelies are really gregarious, especially in the summertime, and they will gather in large rookeries, as this one images, to breed and rear their young. Most of the adults you see here lying on their tummies probably have a pair of eggs underneath that they're keeping warm. I don't know if the lights in here allow you to see this this individual here in the foreground, which has one egg at its feet. There's a rookery really close to Palmer Station. I like to say it's across the street. It's really across the harbor. You need a boat to get there. We don't have streets. That peak population had 16,000 Adelies. The rookery size has decreased over the years for various reasons. And now there's just a couple thousand nesting pairs. But little sidebar here. This is a dip net full of krill that we scooped through a swarm, and you can transfer that dip net of krill into a standard household bucket, as my little cartoon there suggests. That one bucket of krill weighing 15 pounds would be enough krill to feed about 15 adeles, especially when mom or dad are regurgitating most of that to their fluffy downy chicks. The chicks are very demanding, and actually the parents throughout the breeding season lose about 30% of their body weight, going out to sea to collect krill only to bring it back to feed to their chicks. But that one bucket of krill would be enough to feed about 15 Adelie penguins. So I mentioned that at peak population, this rookery had 16,000. So for that one rookery, you'd need 1,000 buckets. Now, even though the rookery close to Palmer, the numbers have decreased, the Adelies are still very, very common and populate a good part of Antarctica. In fact, their most latest census said indicated there are 10 million. The closed captioning has that last couple of zeros blocked. 10 million Adelie penguins. So that's a lot of krill needed just to feed the penguins. Of course, penguins aren't the only predator on krill. This trio of crab-eater seals probably is hauled out on this ice floe to cool off after working up a sweat swimming after krill. Crab eaters can nip them one by one out of the water column, or their teeth are specially designed so that they interlock and that they can take a big mouthful of water and basically sieve the krill from that water. Well, the crab eaters were hunted for seals in the 1800s for their silvery pelt. And sorry if this gets too graphic. When the seal's stomach was cut open, the sealers thought nothing but crabs was spilling out. In reality, it was nothing but krill. So these really should be called the krill eater seals. But these seals get to be 6 to 8 feet long, about the size of this table, maybe a little bit longer, feeding on nothing but a 2-inch krill. Now, an average crab eater would need 2 buckets of krill per day, 365 days of the year to meet its minimum required day, RDA or whatever that is. And this seal is found only in the Southern Ocean, the waters around Antarctica, and it's the most abundant seal in the world. And the census on this seal is 15 million crab eaters. So that's an awful lot of krill just to keep the crab eaters going year round. But it gets worse for krill, of course, when the humpbacks come down from South America for their feeding vacation. They'll come down for about 4 months to feed and fatten up and then go north to breed. Or they'll come down, they'll escort their calf down to the rich feeding grounds and show the calf the moves. The humpbacks are really amazing to see. They're a member of the rorqual or 3-pleated throat whales, and you can see these pleats right here are now fully expanded as this humpback has come up from swimming through a big school of krill with its gaping maw wide open. When they get to the surface, the enormous tongue lifts to the upper palate and all the water spills out through the finely combed baleen plates hanging from the upper jaw. And the humpback gets a nice big mouthful of krill. Now it's estimated that the average humpback takes 1,000 pounds of krill per day when they come to Antarctica. So to go back to my fun with Photoshop, to feed 5 humpback whales for just one day, you need a dump truck full, a 5-ton dump truck full of nothing but krill. And my whale friends estimate that there's probably 6,000 individual humpbacks who come down to Antarctica to feed on krill every summer for 4 months. So I hope I've impressed upon you how the predation pressure is so extreme on this tiny little crustacean. And that's partly the fascination with this organism, and that frankly what drove a lot of our questions. So let's take a closer look at krill. This is a view of a dissecting scope of krill, and krill are great when they are alive. They're basically transparent, so it makes for really, really easy anatomy lessons. So you can see quite clearly here, I'm sure, the heart right there, and then a spent ovary on top of that. Maybe not. How about the green thread right here running through the length of the abdomen? That's its intestinal tract. The olive-shaped organ up here is the hepatopancreas, analogous to our liver. It's a storage organ for excess nutrients. And then the green triangle just anterior of that is the krill gut. And you're probably wondering what's with all the green? The green comes from the fact that krill are primarily vegetarians. So they use these appendages here collectively called, and really cleverly, the feeding basket. Using those appendages, the krill will swim through the water, literally combing it, and on those fine setae, collecting microscopic plant particles, diatoms, and other types of phytoplankton that are rich in chlorophyll, just like spinach. So that imparts that green color to the krill. So the krill have a great life, assuming they can escape predation. All they do is swim all day in the Southern Ocean and feed. So all that feeding leads to a lot of growth. And being a crustacean, they tend to outgrow their clothes on occasion and have to molt. And I've witnessed it only once or twice, and it happens instantaneously. But all those joints and all those limbs and all those setae are absolutely replicated in the new version of the individual, with the new wardrobe on. And it's just an amazing feat, the amount of energy that molting must take and how quickly it's done. And that, I don't think 3D printing could manage that. Well, all that growth or all that feeding leads not only to growth, but also to reproduction. This is a really, really pregnant or gravid krill. The female's ovary head is now bulging with cells that are destined to become eggs. An average full-sized female can carry about 4,000 eggs in the ovary. And it's thought that if that food source of theirs, phytoplankton, diatoms, are of good quality and good quantity, and for a long enough duration, they can regenerate that ovary 7 times in one season. So there's a potential from one female to have tens of thousands of offspring, assuming 100% survivorship of those offspring. But krill don't have it easy any which way. They've even got this crazy, complicated early life history. So the female sheds the eggs out into the open sea, and they all develop away from mom. And it takes about 3 days. The eggs will sink down into the water column, and in about 3 days, the eggs hatch looking nothing like a krill. In fact, it looks more like a tick. It's just this little oval body with 3 rudimentary sets of appendages. And then they start feeding and growing and molting and progressing through 13 larval stages before they look anything like this last image here, which is a juvenile subadult. So it takes about 9 months for the larval sequence, and so they're laid in the summertime, and 9 months from the summertime is going to take that larva into the winter. And so this is what the most common stage of krill looks like in March. In March in Antarctica is in the autumn, when the days are starting to get shorter. It's really nice at Palmer Station. We finally start getting really pretty sunsets like this. So Antarctica does have autumn colors. We just don't have to worry about breaking the leaves to go with it. But what happens to the organisms in the ocean that need sunlight in order to photosynthesize, and then what happens to all the vegetarians swimming in the ocean who rely on that plant material. And remember, part of the continent, especially the Antarctic Peninsula, part of the Antarctic Peninsula is north of the Antarctic Circle, so there will still be some sunlight. This image was actually taken on the winter solstice one day, so we had maybe 3 hours of sunlight, but that's about as high off the horizon as the sun got. And that's not a very good angle for the light to penetrate anywhere into the seawater, and there's solid ice covering that water, so very little light penetration. So as my krill career progressed, the ship support through the National Science Foundation also progressed to ships that were capable of ice-breaking ships, unlike the Hero was, that could actually penetrate ice during the winter time. So 3 times we went down to Antarctica in the wintertime to try and figure out just how are krill surviving in the winter when there's presumably no food because there's little to no light and presumably no diatoms. So the easiest way to study organisms in the water when the water is frozen is to break a lead or an opening in the ice. And then in our case, we would don dry suits and go underwater to investigate. For safety reasons, we went down on a tether. You can see the rope right here. We called ourselves dopes on ropes. And looking back on it now, that was a really appropriate term because that was really stupid. But we got a lot of really good science done while being dopes on ropes and underneath the ice. So here again, you can see the tether. We always went down with a data book, very geeky scientists, even underwater. In this case, it was a dive slate. And we also would carry a really high-tech sampling device, which isn't visible in this photo. We went down with an aquarium net, like type you'd use in your aquarium at home. And like swatting at butterflies, we would sweep that net across the undersurface of the ice. Ideally, when we got down, we would see this. And I don't know if all these little red splotches translate well back there, but trust me, these are all krill larvae associated with the undersurface of the ice. Might be a little bit easier to see in this image because the larvae are at a later stage here. When we found the larvae, they looked just like adults do in the summertime. They had nice green stuff in their guts. They were putting out fecal pellets. They were clearly eating. And in our experiments, they were eating and growing and molting onto the next stages. So that provided evidence for us that the sea ice is really critical for these little guys, for the young of the year, to subsist and grow and molt and ultimately maintain the biomass of the krill in the Southern Ocean. You might be wondering, what about the adults? Well, the adults were a lot harder to find, in part because we couldn't tow a suitable net from the ship on a steel cable through ice, because ice would probably shear off that cable. But when we did encounter open enough water to put sampling gear down, we would find krill, but they would not be nearly as concentrated as they are in the summertime. And they wouldn't look anything like the summer adults do. There'd be nothing in their gut. The hepatopancreas would be really real pale yellow. And there may or may not be anything in the intestinal tract indicating they weren't eating anything, so they didn't need to excrete anything. And we'd also find adults that would, krill are sexually dimorphic, so they have external sex characters. And we would find some really small females that look like they were fully adult females. And their wet weights would be off. So clearly, some of these adults, after they've resorbed all those goodies that they put into the hepatopancreas during the summer, were resorbing their muscle tissue. You know, they were going on an extreme diet and shrinking. But then once we don't know if it's a daylight trigger or just what triggers adult feeding, presumably when the phytoplankton levels start increasing, the adults start then feeding again and resuming their summertime appearance. So those are just a couple of the really fascinating adaptations that this little beastie has evolved over the years of being in Antarctic waters, and why I think it's such a superb organism. But I mean, we could spend the whole night talking about krill here. But I'm going to switch gears, and we'll leave the open ocean and head to, at this point, more near shore benthic environments. So we're in the pelagic open ocean, pelagic swimming. We'll now investigate some of the benthic or bottom dwelling communities. This is a artist's oil painting rendition of cross section of the Southern Ocean, and I kind of like it because it, as a marine biologist, at least it minimizes the land portion of Antarctica, and it's like there's nothing happening up there. It's just rocks and snow, and the real action of Antarctica is underwater, and underwater communities are surprisingly gorgeous. They're barrel sponges like this, sea stars, tunicates, all sorts of amazing invertebrates. So I kind of gave up my krill career, and I like to say I settled down and became a benthic ecologist. So now I study stuff on the bottom, and with my lab group at the University of Alabama, Birmingham, we focus on marine chemical ecology. You can read here that chemical ecology is the study of chemically mediated interactions between organisms or between organisms and their environment. And you can break it down into a couple of different groups within chemical ecology, but we focus specifically on chemical defenses. And within that, chemical defenses from a predation point of view. And I've got the term secondary metabolites here because the secondary metabolites are what allow an organism to defend itself. And a secondary metabolite it isn't a primary metabolite. In us, krill or hemoglobin is a primary metabolite. Secondary metabolites are directly related to the organism's physiology. A little bit more deeper into that, but in real life, these communities are even prettier than that artist's rendering. Large barrel sponges like this one. Barrel sponges have an incredible diversity of morphologies and colors in these communities. And personally, I think in some areas that diversity in color rivals that of what you would find on a coral reef. There also, in addition to invertebrates, there are a lot of seaweed in Antarctica. I'm talking macro seaweed, macroalgae, macrophytes. I use those terms interchangeably, so I hope I don't confuse you. So algae everywhere in the world are color coded. You've got brown algae, red algae, and green algae. And Antarctica has all three of those as well. Some of the brown algae are towering, not unlike the kelp out in California. There are, and we call these Antarctica's trees. There aren't forests in Antarctica. Well, there are forests in Antarctica. They're underwater and they're with algae. So these big browns are the trees of the forest. Then there's a huge diversity of smaller, more delicate red algae that kind of form the shrub layer underneath the canopy of the browns. There's also a couple of species of green algae, and it's always kind of refreshing. There's one species that can form just like this nice little yard or stand of green, and it's, oh, got some green here in Antarctica. It's a really nice, fresh, refreshing respite from the otherwise brown or black, white, and blue up top side. And here's just a really pretty, I think, kaleidoscope of brown algae here. Here's a little mini meadow of that green alga, and then numerous species of red algae over there. Some of these big brown algae don't stand upright, but rather lay horizontal across the bottom. Often in some areas, they will cover 100% of the bottom. One species in particular, Hemanthothallus grandifolius, it's huge. Its lasagna noodle-like thallus wraps completely around these two 6-foot-tall plus graduate students. I think the record total length for this particular species is 50 feet, so that's a lot of biomass. The other dominant browns in the communities are more bushy. Two species that look an awful lot alike and confuse even experts sometimes. All right, so I should have prefaced this section by saying I want to introduce a different group of crustaceans. These are amphipods, a relative of krill. Amphipods are sort of described as the insects of the ocean, in part because there's so many of them. But it's not like mosquito insects, you know, that bite you. These are typically very beneficial, and I'll explain that in more detail in just a second. But the amphipods, while there's only a handful of krill species in Antarctica, there are hundreds of species of amphipods in every single niche you can imagine. And collectively, they can be as abundant as krill. Now, there was a graduate student doing algal amphipod interactions in our lab. He was quantitatively sampling 2 different species of algae and then quantifying the number of amphipods on those algae. And on one species, he would routinely find in the tens of thousands of amphipods on that particular species of brown alga. On another one, 300,000 individuals of amphipods. Now I've got the units there as meter squared. So meter squared is like 3 feet by 3 feet, about the size of a rose bush. Imagine if you had 30,000, much less 300,000 aphids on a rose bush, or that many caterpillars on my local sentimental favorite shrub is lilac. So a small lilac, if it had, you know, that many caterpillars on it eating its leaves and flowers, you'd be going nuts. But in this case, it's really beneficial for these algae to have these amphipods associated with it. We've done lots and lots of experiments after some of those early observations. You know, Louis Agassiz, I think it was, said study nature, not books. Don't give this one to your professors though. And really a lot of our work stemmed from, did you see what was happening, with that? So we observed something and then we questioned it and followed up on it. And as a consequence, we discovered that there are an awful lot of amphipods that live on algae but don't eat particular algae. But they'll eat some other algae but don't live on it. And we also noticed that there was a similar thing going on with fish, that fish would eat amphipods off some algae, but not off some other algae. So it turns out Antarctica is a really great place to study chemical ecology because, and I'll read that line, most macroalgal species or seaweeds are unpalatable to sympatric consumers. So a sympatric species are two different species who live in the same habitat. So in that previous slide, the amphipods and the shrimp, and the amphipods and the fish are sympatric to each other. And unpalatable means, it doesn't taste good or it's not nutritious. And the majority of the macroalgae in Antarctica fall under that category. They're unpalatable. All of those, the dominant browns, and the dominant browns are the three that I introduced you to, are unpalatable, and a lot of the common red algae are unpalatable. So we came to realize that there's some mutualism going on between at least the amphipods and, sorry, I've got macrophytes here, macroalgae, again, the same thing. The macrophytes, through that chemical defense, are protecting themselves from herbivory by the amphipods, but they're also giving the amphipods a refuge from that fish who think the amphipods are really tasty. So it turns out that it's kind of a win-win situation. The macrophytes, the way I've got it set up here, the amphipods win by associating with the macrophytes or the algae by gaining a refuge. And the amphipods perform a wonderful service to those macrophytes. They don't eat the algae, in part because their mouth parts are too small, and in part because the algae don't taste good even when we extract out those chemicals. But they do eat the epiphytes, and epi, to dissect that, epi means on, phyte means plant. So the amphipods are basically keeping the thallus, or the blades of those algae clean and free. And what do algae are basically plants, right? They solar collect for a living, and you've got to keep those solar panels clean to keep them at maximum efficiency. So that's what the amphipods do for the macrophytes in return for a refuge from predation. So it's a win-win situation. And we came to realize that it expands beyond these two groups of organisms. We've done a lot of work with sponges and tunicates, to mention a few invertebrates, and see that they're similar situations. So it's community-wide mutualism. Now, one of the principal investigators is a natural products chemist. So whenever we observe this curious interaction, he and his team use their chemistry magic to determine, to literally boil down the organism and extract what that deterrent chemical is. And we'll use that in our experiments. And the chemistry guys will send off the purified compounds to various agencies like NIH for potential use in the medical field. And from one of the red algae, it's now been renamed, has a protein that in some screening trials had effectiveness against against influenza. That one hasn't really gone anywhere, but what has caught an awful lot of interest most recently is from a sponge. This yellow spiky structure here is a sponge called Dendrilla membranosa. It's a member of the Darwinolide family of sponges and has this unique compound that they term Darwinolide after the family name of the sponges. It's really effective against MRSA, the biofilm MRSA. And MRSA is that hospital-acquired Staph aureus that's really, really hard to deal with. And there are very few things out there right now that can deal with the biofilm version of MRSA. So this is somewhere along the pipeline in clinical trials. Very exciting. And one more example from a tunicate. Now, a tunicate or sea squirt is an invertebrate, a very distant relative of ours. They're chordates, like we're chordates. The one thing chordates all have in common is a primitive notochord at some point in their development. So these guys have it in a real early life history stage and then lose it. Anyway, this particular tunicate, Sinoacum, produces a compound that was called palmarolide A after Palmer Station. And it's incredibly effective against human melanoma, but so far only in cell culture. But it too is somewhere along in the clinical or the pipeline to clinical trials. So it's not at all a major part of our project, but it's really exciting when basic science can have an impact and an application to better the human condition. Okay, so we're going to switch gears again, go a little bit deeper, and I'll include another crustacean group. And this one's going to take a little bit of setup, so stick with me. Probably heard that Antarctica is one of the most warming places in the world. This graph nicely illustrates that. I'll walk you through it. On the vertical axis is the average yearly, or the average winter air temperature, so June, July, August, air temperature versus decade. The black data is from Faraday Station, which is about 60 miles south of Palmer Station along the peninsula. It's a British station. Well, it was established as a British station in 1945 and recently was taken over by, or changed hands. It was a very friendly takeover by the Ukrainians. It's now called Vernadsky. But anyway, they kept all these air temperature records. The red lines, the red data is data from Palmer Station. So these are average winter temperatures from 1945. Palmer jumps in at about 1970. The doors of Palmer Station opened in 1965 to science. So the general upshot of this graph is that the slope is going up. So air temperature is rising with time on that horizontal axis. And pretty much about 1 degree centigrade per decade, and that's air temperature warming. 60 degrees over what is that? 6 degrees over a 60-year lifespan. So that's significant for an area where it's the organisms at least are stenothermal, used to a very strict non-ranging temperature limit. But warming air temperatures also have ramification for warming water temperatures. And this chart depicts the extent of annual sea ice. That annual sea ice I showed, that ring of ice around the continent. And just the general shape of the graph. So you've got the months of the year here and the sea ice area on the vertical axis. So it's, not uncommon for the summertime for the sea ice to be at its minimum. So it's the lowest in January, February, and March. It's starting to build up again. Peaks in July, the winter time, and then starts dropping back as spring hits and summer starts up again. The gray lines, we'll dig a little deeper, the gray lines are all kind of a mash of the years between 1981 and 1980, or 2010, sorry, with the black line being the average of those years. The blue line is the year 2022, quite a bit below that three-decade average, and then 2023 is coming in at a record low, even lower than 2022. And the warming water, and we know that from the krill larvae how critical that annual sea ice is for maintaining the populations. The warming water has other consequences for these shallow subtidal organisms, and it comes from the real deep sea. Now, in part because it's so cold in Antarctica, there are no crushing predators. You don't find sharks or turtles or parrotfish. None of the fish are heavily jawed. And it was thought that there are no crabs in Antarctica. And as a consequence of that, the organisms that live in Antarctica that do build a shell, specifically mollusks, snails, clams, oysters, don't have really heavily calcified shells. It's really hard. The cold water makes it that much more energetic process, and there's no need for the organisms to heavily calcify if there are no crushing predators. But with advances in technology over the years, as this little cartoon suggests, scientists are now able to use remote vehicles or towed vehicles to explore deeper into the Southern Ocean. And sure enough, there are crabs. But they're crabs that are limited to the deep sea. Crabs, if the water temperature gets too cold, they tend to get narcotized, like they're drunk. They can't handle all that calcium. They can't clear the calcium as well. And they'll basically get paralyzed, won't be able to move their gills, and they won't survive. So there are crabs in Antarctica. They're just down in the deep sea where it's a more stable temperature and doesn't get below 0.5 degrees or so, which the surface waters clearly do, especially when there's sea ice on top of it. So I participated we had a series of 3 cruises over the years where we towed a camera array like this across the seafloor, hoping to document the distribution and abundance of these crabs, and hoping we got any crabs. Well, the first time we put the camera down, boom, we got crabs. There was a live feed from the instrument array, so we could watch in sort of real time. So it was really exciting. We collected hundreds and hundreds of images of crabs like this in lots of different stages of their life history, lots of different interactions with other organisms down on the deep, dark sea. We found some really curious interactions. We found teeny tiny little crabs hanging onto sea stars like this. We call those crab cabs. Don't know if it's the lazy crab way to get around on the sea floor, or if they were going to help themselves to whatever food the sea stars managed to find. So many questions. On the third and final cruise, we attempted and obviously succeeded in trapping crabs down 1,000, 1,015, 1,500 meters. And we're really pleased when the traps came up with 50-some crabs. Mostly they were males, but there were 9 females, and of those 9 females, 3 were egg-bearing. And so evidence that it's a reproductive, established population. We gleaned as much as we could out of those live specimens, including looking at what they were eating. So I think this is my last graph. This is the frequency of whatever they had been eating, and whatever they had been eating is on the horizontal axis here. Ovuroids are brittle stars, were the most common. Echinoids are sea urchins, mollusks, clams. Asteroids are sea stars, cucumbers, sea worms, rhizomes, and sponges. Primarily the ovuroid, the brittle stars, and the echinoids seem to be their go-to meal. And in the inset over here is a photomicrograph of a smear of gut contents. And you can clearly see part of an urchin spine here and an urchin test over there. A lot of the other white structures are brittle star bits. Now we thought these crabs must be pretty lethargic and not capable of taking live prey, and that they must have stumbled across dead brittle stars and dead urchins because they couldn't move quickly enough to catch any live prey. We really couldn't determine what the state of the prey had been once it was in the gut. One thing we were able to determine is, yes, they are crushing predators. I twice mishandled a crab, and I kid you not, it took 2 people to pry my thumb free. They're very, very strong. So no doubt about crushing predators. And this was mentioned in the introduction, but years ago I had the most phenomenal opportunity to be a participant in a submersible survey of Antarctica's sea floor. And came face to face one day with a couple of crabs down at depth. And this poor fish got a little too close to this crab, and the crab, lightning speed, snatched that krill, snatched that fish right out of the water column. And then the crab proceeded to run away from the other crabs who wanted a piece of the action, I think. And the submersibles are great. They go up and down really well. They do not spin. They do not go horizontal. The pilots, had that sub revving, trying to keep up with this crab that was running away from us. So, evidence that they are both capable of taking live prey and that they are very, very agile. Now, I bring up these crabs because there is concern, of course, that as the surface waters, if they continue to warm, there will no longer be a thermal barrier, and these crabs will be able to mosey on up into the shallower waters where there's a heck of a lot more stuff to eat. It's not, the organisms are not protected, they're mostly soft-bodied, and they're abundant compared to the sparsely populated deep sea. So time will tell on what will happen with the urchins and the anemones and the brittle stars and the sponges. I don't know, maybe one of you students here or out on Zoom someday will be able to report back that, yeah, Antarctica's ecosystem is nice and stable and the crabs are down in the deeps where they should be. And I'd like to close with another submersible experience. As a krill biologist, I was really, really pleased one day. We were in a bay that was full of humpback whales and they were feeding on krill, but They were way over there, and we were operating very far away from them. Obviously, must have been a very large krill school. I had seen krill on my previous dives, maybe one or two here in the water column swimming by. I'd never seen a big group of them. And they were attracted to the lights of the submersible. The purpose of the submersible cruise, it was a media cruise, so they were filming a documentary. So there were lights and cameras and lots of action, especially out there was lots of action and even more action. At one point I remember remarking, you can't even tell there's water out there beyond our 6 inches of plexiglass. It was just nothing but krill. I was obviously enthralled and actually a little bit overwhelmed. And two things went through my mind. One was that there are whales just over our heads, possibly, and is this going to be a Jonah experience? You know? We can't see anything. Do the whales know we're here? And seriously, the other thing I thought about was Dr. Mary Alice McWhinnie and how I wouldn't be sitting in that submersible 400 some feet down in Antarctica in a bay filled with krill being chased by humpback whales. It was just, no pun intended, but a very profoundly moving experience for me, which I never would have had it not been for a special educator here at DePaul who really set me on my career and my life trajectory. So I never not give a talk like this, especially to students, and not acknowledge Dr. M.A. and her impact on my life. The biolabs at Palmer Station are named for Mary Alice, and I go by this every single day when I'm in the field, so it's impossible not to think about her and to think, even if I'm having a crummy day, it's like, thank you, Dr. Mary Alice, this is all because of you. But I would not have been there had it not been for her. And I really, truly hope that someday each and every one of you students can look back and say, gosh, whether it's an educator, a friend, a family member, you can look back and say, and identify one particular person in your life who just made your life. And that's what Dr. Mary Alice did for me. So I'd like to acknowledge a whole bunch of people. I use the royal we a lot. There's no I in research. I've been fortunate to work with a lot of great institutions, a lot of great funding agencies, and field teams that have really helped. You know, you don't do it. It takes everybody on a team to do science, particularly in Antarctica. Now look closely at most of these images though. There are an awful lot of women in these images. This is not Dr. M.A.'s time. Women are in Antarctica and making an impact in science and also in support. I want to draw special attention to this image over here. This was taken last March on International Women's Day. That's almost all of the station female component. Now, Palmer's only 44 people at max, and I don't think we were at max population at that time, but there were close to 20 women on station. At one point, it was just myself and Dr. M.A.'s postdoc who were the only females at Palmer. So a lot has changed in Antarctic science. It's no longer the male-dominated bastion that it had been for so many years. And about 50% of these women are science, 50% are support staff. And Palmer's basically a little village. You know, we've got everybody that you would have to keep the station running. We had the power plant one year was the power plant mechanic who kept the generators running, who kept the lights on and everything, was female. This is the admin coordinator who manages all these station operations. This is the boathouse coordinator. She is a licensed boat captain herself. She can tear down and put back together an outboard motor in no time. Amy is the logistics expert. She drives all the heavy machinery, the front loaders and the mobile cranes to move massive bits of cargo around, the biolab manager. You know, my point with this is whether you're science or support, or in this case, a supporter of science, because everybody at Palmer Station is there to support science. That's the reason the US has those research bases. Find your passion and run with it and never be denied or held back by your gender. So thank you, and I'm happy to take any questions.